Yeah, sorry. Welcome to another session of Bioinformatics. I am Dr. Y.V. Rajesh, Assistant Professor, Department of Biotechnology, KL University. So in today's session, we will learn about transitions, transversions and nucleotide substitution models that are used in bioinformatic phylogenetic tree constructions here. So the goal of phylogenetics is very simple. The molecular phylogenetics, which uses DNA or protein sequences, its goal is very simple here. It will ask several questions here. So what is the history of life on this planet Earth here? So can we tell the history of life on this planet Earth? So was the extinct quagga, an extinct animal quagga, is more like a zebra or like a horse here? So it looks half evolved from the zebra and half evolved from a horse, but by using simple sequence element and molecular phylogeny, we should be able to address this question here. So was Darwin correct that humans are closest to the chimpanzees and gorillas? So we can ask this question. Then how related are whales and dolphins, horses and to cows here? So which are the species? Whales, dolphins and cows, which of them are more close together? And we can ask questions and also address the question, where and when did HIV originate, and how many genes are related to my favorite gene here? So these are the questions that are asked in molecular phylogeny. So molecular phylogeny can address all these questions with the help of sequence data and bioinformatic softwares and interpretation using experts, bioinformaticians here. So the approach, the bioinformatic approach to resolve these issues is basically characterized into phonetics and cladistics, the phylogenetic tree constructions here. So phonetics, basically in classical phylogeny. So we will try to classify organisms based upon overall similarity and overall morphology and other variable traits here, regardless of their phylogeny or evolutionary relations here. So same approach can be extrapolated to sequences also. So we will look into overall sequence similarity of sequences and we can construct the phylogenetic tree here. And next approach is cladistic approach, where biological classification can be done on the shared derived characteristics. So here derived characteristics is very important here. So that means they must have some common ancestor here. From a common ancestor they might have derived here, and based upon that one you try to construct the branching patterns here. So the phonetic approach, generally we call them as character based approach, and the cladistic approach, generally we call them as a distance based approach of phylogenetic tree constructions here. So before learning about these methods, so we should learn about there is something called as a matrices, substitution models for phylogenetic tree constructions. So we know that during the evolution the DNA will undergo lot of mutations here. At the nucleotide level there are silent missense, nonsense, insertions or deletions will happen. At genomic levels amplifications, translocations and inversions, interstitial deletions will happen here. So the events of at the nucleotide level, the mutations basically depends upon the characteristics of the nucleotides which are present here. So all nucleotides doesn't have same substitution or mutation rates here. So mutation means one character is changing to another character here. So DNA we have 4 different types of nucleotides: adenine, guanine, cytosine and thymine here. So out of this adenine and guanine, those are called as purine nucleotides. Cytosine and thymine are pyrimidine nucleotides here. So we assume that A can mutate to C, A can mutate to G, or G can mutate to T. It's same rate, maybe normal assumptions like that, but if you see they are chemically different. The chemical structure of all these are different here. Adenine and guanines are more similar because they are purines. Then cytosine and thymine are more similar, they are pyrimidines here. So here transitions, we define transitions and transversions. Transitions is purine to purine mutation or pyrimidine to pyrimidine mutation are called as transitions. Transversions, if a purine is changing to pyrimidine, that means adenine is changing to cytosine, or guanine is changing to thymine, or guanine changing to cytosine, we call as a transversions here. So there are 4 transversions and 2 transitions possible for 4 nucleotides here. So basically, with this knowledge, scientists have developed models of nucleotide evolution here. So those models have been applied for phylogenetic tree construction. The initial model, the first model which was developed is Duke and Cantor model in 1969. So this model assumed that the rate of substitution is same for all nucleotides. Maybe they may be chemically different here, they may be chemically different, but rate of substitutions or rate of mutations amongst the different nucleotides will be same here, Duke-Cantor model. But with some data availability after 1969, so 1970 a new model has been developed, model of nucleotide evolution, that is called as Kimura 1980. So it found that there is some difference are there here. So the rates of substitutions is not same, so some difference are there here. So the substitutions is the transitions and transversions are not same. So transitions occur more frequently compared to the transversions in nucleotides here. So the Kimura 1980 model, it assumes that these two are completely different parameters here. So we should consider different weightages to them here. So based upon these ideas, so Duke-Cantor model, it uses something called as alpha, value of alpha, that is substitution rate, assumes that all are same here, transitions and transversions are same and same score will be given to them. Whereas Kimura model, for 4 transversions, beta a higher value and 2 transitions, alpha smaller value. So beta is greater than alpha used in phylogenetic tree constructions here. Okay, so in phylogenetic trees here, there are 2 types of trees are there, rooted tree and unrooted tree here. So any tree will have whether it is a rooted tree or unrooted tree, any tree will have leaves, branches and root here. So root is the starting point or origin. So from this one branching will happen here. So these are called as branches, and from branches there can be another branch points and sub branching will happen here. So the branch point is called as internal node, and the final taxa are called as, simply final things are called as taxa or terminal nodes here. This is a bifurcating tree, that means it is going to 2 directions here. So a tree can be trifurcating or multifurcating. So this is a trifurcating tree, or if you have more than one thing, that will be multifurcating trees here. So taxa or terminal nodes, branches internal nodes, and roots are the features of a phylogenetic tree here. And unrooted tree is when we don't know where is the root here. So where is the root or where is the ancestor, then that will become unrooted tree here. So I have A, B, C, D, 4 different taxa are there. We don't know the tree here. Maybe this will become unrooted tree here, whereas it is a rooted tree here. So the number of trees possible for based upon number of taxa we have is given by some formula here. So number of rooted tree is given by 2n power minus 3 factorial divided by 2n minus 2 into n minus 2 factorial, and unrooted trees nu 2n minus 5 factorial divided by 2n minus 3 into n minus 3 factorial here. So these formulas indicate that, so even if you have few number of taxa also, there are many number of trees are possible. So the construction of optimal tree is a computational problem in bioinformatics here. And in this session we learnt about transitions and transversions and models of nucleotide evolution and some definitions of rooted and unrooted trees. So thank you for listening to this session. Thank you very much.